Hello everyone, I'm Annissa, and today I'll be discussing the effects of AI and nanobiotechnology in the future of pharmaceuticals. So I thought that this would be interesting to look into, since throughout the course we've been talking about how AI could help with diagnosing patients with illnesses. So I wanted to see how AI and nanotechnology could also improve the treatments that people would receive once they've been diagnosed, and even how these technologies will be useful once the conceptual shift in medicine occurs from treating patients' diseases to enhancing patients' lives. So the outline for my presentation will first begin with a brief introduction of AI and the different subcategories of AI used in pharmaceuticals. Then introducing what nanobiotechnology is. Then I'll be talking about drug development, drug delivery, and the challenges we currently face in these areas. And finally, I will conclude with current technologies such as AI Cure, IBM Watson, and particle robots, and how these can possibly be used in the future. So artificial intelligence is the simulation of computers to mimic human behavior. There is a subcategory of AI called machine learning, which uses algorithms to learn without being programmed to specifically do a certain task. Within machine learning, there are 4 subfields: supervised learning, unsupervised learning, reinforced learning, and deep learning. Supervised learning is the use of classification and regression methods to make a predictive model based on both inputted and outputted data. This means that outputted data from supervised learning would be from classifying data into subgroups, creating a predictive model, then using this model to predict outcomes for new data. Unsupervised learning is the use of grouping and interpreting only inputted data. From this, new data will be clustered, and certain features will be used to filter through the data. Reinforcement learning is based on giving the machine a goal to maximize its performance so that it can make a correct decision in a given condition. Outputs given by this method would be outcome-based, where the option provided is the best possible decision to maximize the goal that was originally set. Deep learning utilizes artificial neural networks, which try to simulate the neural networks in the biological brain. It is composed of layers of interconnected units and can adapt and learn from experimental data. So all these subcategories can be used with big data, data sourcing, and data mining to help pharmaceutical companies develop new drugs. So next I'll be introducing nanobiotechnology, which is an application of nanotechnology, the multidisciplinary field that manipulates the size and shape of matter at the nanoscale to biology. So as a good point of reference, the size of a tennis ball is like 10 to the 8 nanometers, and the diameter of human hair is 10 to the 5 nanometers. So nano devices that can be used in medicine, or nanomedicine, as you can see in this picture, is that they're extremely small. Like, they're like what, 10 to the 1, 10 to the 2, right? So they're like way smaller than like the diameter of your hair and stuff. So we can't even like see this size like with our naked eyes or anything, but maybe in the future, if our eyes get enhanced or something, we'll be able to, but for now we can't. So anyways, nanotechnology can be applied in treatment, diagnosis, and monitoring of diseases. The size, solubility, mobility, and surface area that nanotechnology can provide can be utilized by pharmaceutical companies to design novel formulations to be used in healthcare. And as a fun statistic, it is estimated that the nanomedicine market will be continually growing by 12.5% annually between now and 2023. So this suggests that nanobiotechnology will inevitably be integrated into medicine and used as a tool in healthcare in the future. Drug development and design. Drug development is the formation of a drug that can be used to treat diseases or illnesses that a patient has. Before a drug can be created, a lot of research and planning needs to be done to see what materials can be used to synthesize a new drug to have an effect or an effective effect on a biological target. So obviously they can't just be like, oh, like this chemical kills cancer cells, so let's make it into a pill and sell it to people, right? Like, that's not how it works. So we need to know the mechanism that the chemical uses, so to see if this can even work in our bodies, such as complex, which is such a complex biological environment, and see if the side effects, or if it has side effects and if it's even toxic to our bodies, right? So a lot of research needs to be done prior to being able to sell a drug. So first we'll be talking about the forward approach of drug development. So the first few steps in drug development is the identification of a compound that causes biological activity. Now these compounds are called hits, and hits are usually natural materials isolated from plants, or bacteria, or fungi. And basically they tell us that this specific compound causes some sort of activity in the cell. For example, this change in activity could be slowing down the growth of tumor cells or something like that. So once this has been identified, the next step would be identifying chemical compounds or leads that have potential to be synthesized into drugs that have maximum therapeutic benefit and a minimal harm to the patient. After the hit and lead are both identified, they need to be tested extensively in biological assays, in vitro studies, in vivo studies, or animal models that accurately portray the disease state so that the safety of the drug can be determined. During this process, the hit and lead may be modified for increased specificity. Oh my gosh.
Specificity.
Yeah. Or therapeutic effect. So this is kind of like educated trial and error, where you keep testing if changing a side chain here or there will give you a more desirable effect or not. So yeah, moving on. Drug delivery is the transportation of the drug to its targeted site. This is really an important step as well during drug development because how the drug is delivered affects its efficacy in treating the problem. So before the drug can even be delivered from one place to another in your body, there are many different ways that the drug can be administered. For example, medications can be administered orally, through injections like IV, so in your veins, intramuscular, through your muscles, through the skin, topically or transdermally, inhaled, and stuff like that. So these different administration methods allows for the medications to be delivered in specific ways. For example, if the active ingredient in your medication can't first pass metabolism, as in it'll get degraded by your liver or in your stomach, then taking the medication orally might not be a good choice because the drug won't even get to its delivered target site anyways, so it won't have any effect. As well, the different conditions the medication faces during its delivery may change how effective it is as well. And being exposed to different changes in pH and being in the presence of enzymes degrades some of the drug. The amount of drug available to treat the affected site will be less by the time it reaches it. Also, if the drug is too bulky or it can't bypass certain biological membranes or barriers to even reach the site, again, how can it be effective if the drug can't be delivered? So these are all questions that need to be answered during the drug development and design process. So ultimately, all the challenges in drug development that I've read so far leads to increased costs in research and development, both from explicit and implicit costs. Explicit costs being the actual money spent, and the implicit costs being the time spent on research. So as we can see here, this graph kind of looks like something we've seen before, like sort of like Moore's law, but not really, because so we can see that over the years there's been exponential growth in the cost of developing drugs. So I think this is pretty scary that the cost of drug development is rising so quickly, especially if the patients are the ones who have to carry part of this hefty burden in the end. So especially since the increase in cost of drug development isn't linked to increased output of newly approved drugs either. So it's like so much money spent but not that much output created. So there are many factors that contribute to this research and development inefficiency. First, it's the desire to replace drugs on the current market with newer and improved drugs that have better pharmacological activity. Essentially, this causes pharmaceutical companies to invest more time and resources into finding better drugs that treat the same kind of disease. This is good once the drug has been developed, because we'll have better drugs to treat our illnesses, but at the same time, as we continually try to improve something that's already good, like it's hard to find better solutions, right? Because you've exhausted like all your possibilities, kind of. So that's why it takes exponentially more time and more money with each round of improvement. Secondly, following the same kind of idea as the first, we want to be able to decrease risk tolerance and increase the safety of the medication. But this comes off as a trade-off with cost, because the longer the company has to spend on research in these novel solutions, the more inefficient research and development becomes. Finally, the current brute force approach that many pharmaceuticals use to screen for targets are often too simple to look for new, complex, and unique targets. Usually, drugs that interact with multiple biological targets will have greater therapeutic effect. But if the screening process is too simple, then oftentimes we'll just miss these better targets for what we're trying to create. So in addition to these challenges, there are also challenges during clinical trials, such as inappropriate study designs, and if there's incomplete understanding of how the disease and the pathways and mechanisms are involved, if that's not well understood, then the study may be designed to ineffectively test for the efficacy of the drug. So maybe instead of a causational study, it's a correlation study instead. Alongside this, it's important to have a good selection of patients to participate in the clinical study. If the patients chosen are not suitable, it can affect the results of the study and may cause the study to fail. So AI can help with the challenges that I just mentioned. IBM Watson for drug discovery was previously used to identify new RNA binding proteins that were linked to a neurodegenerative disease. Its ability to complete the pathway of how the disease worked helped researchers determine appropriate study designs for the clinical trials. Machine learning and various predictive models can be used to identify target-specific compounds more efficiently to maximize safety and efficacy of the compounds. The efficiency will help reduce the cost of research and development. Deep learning can also be used so that researchers can fill in gaps when there's insufficient data on structural information on compounds using biological or molecular networks. Another way AI can help is with the selection of patients for clinical trials and monitoring the progress of the study. AI Cure, which is a novel AI platform, measured medication adherence for patients suffering from schizophrenia in a phase 2 trial. And it was able to increase adherence by 25%. This is great because if patients don't adhere to the treatment or medication, it could really skew the results of the study, especially if you're trying to test if a drug is working or not, but the patient doesn't even take the drug, so how will you even be able to test it, right? So, in a more in-depth look at AI Cure, from the company's website, they claim that it's an AI and advanced data analytics company targeting the healthcare industry. Their proprietary intelligent software captures and understands video, audio, and behavioral data to establish the link between patient's disease and treatment. I actually watched their promo video on this app, and how it works is basically it reminds patients who are in these clinical trials to take their medications on time. As well, it documents and verifies that they've been taking their medication using facial recognition. Essentially, they detect the pill, like you show your like phone the pill, and then you like kind of like selfie and like show that you're like eating it. Essentially, it like detects that you've actually consumed the medication and then sends the data back to the researchers and healthcare professionals. This is pretty cool because it provides live feedback on both the patient and researcher's end, and it can also detect any questionable behavior. So in a way, it helps researchers find suitable candidates for their clinical studies, as they will have this app to monitor their patients, and they can really see if the drug being studied is being effective or not, without worrying if the patients are being dishonest or like forgetting to take the medication altogether. So yeah. So challenges in drug delivery. So in order for a drug to be effective, it needs to be able to be delivered to its target site while maintaining enough of its therapeutic effect to treat the area once it's been reached. So taking the example of cancer cells again, as you can see in this photo, that there's a lot going on in this tumor, right? There's cancer cells, cancer-associated fibroblasts, macrophages, endothelial cells, extracellular matrix. So these different cells may all have slightly different environments in their cells. So as well, tumors have different cellular stresses, such as low pH or hypoxia. So any cancer treatment to tumor cells have to be able to be effective in these harsh conditions, which makes it challenging. As well, in cancer cells, chemotherapies are highly toxic and potent. So ideally, drug delivery should be specific to tumor cells so that other healthy cells in the vicinity don't get harmed. However, oftentimes drug molecules diffuse freely throughout the body, so highly specific and controlled doses may be more diluted once it's gotten to its target state because they've diffused elsewhere. So physical characteristics of the drug is important as well. If the size of the compound is too large to bypass a membrane, it might not even be able to reach its target, like I said before. So if it can't reach its target, then it won't be able to do its job. So this is where particle robots and nanotechnology could help. Before I get into particle robots, I want to mention that currently there are several drugs in development for cancer that are based on nanoparticles. And approximately 150 drugs in development are based on nanotechnology.